more of a general theme of a contrast mechanisms in optical microscopy. So I rearranged it. It's probably better to give you homework at the beginning so that you can see the focus, the motivation, because the homework is a very interesting one, the pinhole camera and phase contrast and dark field microscopy. And this is a part of our class material. So once you go through with me today well enough, then I'm sure you will have no problem solving this problem. So the goal is easy, but how many of you have actually seen the pinhole camera? Raise your hand. Okay, good. Sadaf. But the others probably not. It's a very easy, but it's a very, very fun. So while we are doing this, it would be very interesting to understand, hey, there's a pinhole camera, how does it work? And that's something I want you to think about. I mean, you can find solutions in the web, but I would consider you to think through yourself, because that's what matters, to figure out how pinhole camera works. And based on the knowledge in today's lecture, basic principle, and also the effect of pinhole size on the resultant image. And the second is actually part of the lecture material, but you want to think a little bit through. So the goal is, you guys probably have heard phase contrast microscopy, which got Nobel Prize. We'll deal with this. And how to improve the contrast of phase contrast microscopy from your ingenuity, that you can think of it how to improve. So actually I brought the phase contrast microscope part. Remember last time when I brought the pathologist microscope that actually has a phase contrast, and we'll get into this today further. And also dark field microscopy. And it's a coincidence that last week, our departmental seminar is actually given by Professor Zhu from Yonsei University. He's the expert of creating all of this with a fun LED illumination. So some of you remember that? That would be very helpful for today's class. So I give you about like 2 weeks, so plenty of time. But what I want you to think is that you think through yourself. Also at the same time, collaborate with friends who are taking the same class in your lab or another lab. I want you to discuss, okay? But write it down. I discussed this one with, let's say, Jungmyeon. It's very important. So that's kind of a rule that you're free to discuss, and I encourage, but do not just copy the others one. When you discuss, it's totally encourageable. But when you write it down, do it your own way. So that's how I think you can learn and you can develop your own ways of theory. Okay, so today's outline is because we are trying to understand further in terms of microscopy techniques and imaging. And the history of microscopy is really about developing new ways of improving resolution and come up with a new contrast mechanism. So that way, I think it's worthwhile to cover phase contrast microscopy, dark field microscopy, and for understanding this further, there's a type of microscope contrast mechanism based on polarization. So we will talk a little bit, and then DIC, which is also well commercialized, and you can find it many of the cell dealing laboratories, so-called DIC, or differential interference contrast microscopy. Before then, let's try to consider about transparency film. How many of you have actually experienced transparency film? So transparency is something like this. This has some color, but do you remember in the past, maybe 10, 20 years ago, people, or in the lecture, people printed out and then put this on transparency and then make an image onto the screen. Have you ever seen or experienced this? Raise your hand. Okay, good. Only Zhengmyeong. Oh, Sada, great. Yeah, you know what? That transparency, you can consider it as kind of a transfer function. You know, like old days professors, when I was taking lectures in my graduate school, still, I think we used to use transparency. Okay, so let's decompose what the transparency is about. So let's say you are shining with a plane wave, you're going through, and you hit the transparency. And how do you consider transparency as it based? When we printed out letters and figures on top of transparency, that will look like this dark. So that certainly has a amplitude aspect. In general, the thicknesses are kind of similar, but there are cases, for example, let's consider transparency. There is a cells growing on top, and the cell has a special refractive index, which is different from the air. So the thickness of the cell will affect in terms of phase changes because the refractive index are different. So in that regard, you can also consider, let's say, this thickness as kind of representing the physical thickness, but it will affect on the phase status of the light wave. You know, the wave has an amplitude and phase. Okay, and last time we discussed about the complex representation, saying that this plane wave or wave is the amplitude times e to the j and the phase function. So in that regards, this is more like practical aspects. Hey, the thickness might not be that thick. I assume that this one's like 50 lambda is a lambda considering like, let's say green light about 500 nanometer or 0.5 micron. And you can see this one will be less than about 25 micron. If you consider about a cell, single cell size is cell body is about 10 micron or so. So this is completely mimicking, or we are considering even there is a monolayer of cells, this would completely satisfy those. And the lateral dimension of this, you can consider anyway, this lateral, it looks like a pixel, will be similar or bigger than lambda. So that's kind of a constraint we are opposing in this condition, because when it becomes smaller than lambda, I think you would suffer from much of a diffraction, which means the directionality of the wave will deviate. And let's not consider that. Just so this lateral size is big enough that the overall wave is still propagating in the same direction. However, so we can consider this as a, now let's think it as more mathematical aspect. So what would be the wavefront like? So when it passes through, you can see these parts will move forward, but these parts have a more thicker, which means usually this has a higher refractive index, so it will be retarded. So it's a little retarded than the other. So you can see the wavefront now can be a curved, not that much though. This is only a kind of approximation. So in that regard, we can mathematically describe this as a wave after the transparency. Let's put that as a g of x, and that this transparency, let's define it as a transparency function of t of x, and the input as f of x. Okay, so here f is a plane wave in Zhengmyeong. Can you describe this plane wave fx in terms of any function? An amplitude. A plane wave.
Different word, you mean?
In mathematical aspects.
Sinusoid.
Yeah, sine, cosine wave. something you remember last time. So remember when we converted into a complex, important thing is this plane wave, I wrote it f sub x, but in fact, there's always t, and there's exponents I e to the j kx minus omega t is a plane wave. Remember that? And but I said minus omega t is always there. Let's take it off because it's always there, right? Then e to the j k x, and that's what what's over there. And but the amplitude was 1. So amplitude is a constant for the plane. So that's why the simplest form of this is, hey, plane wave gets in, that function multiplied by transparency, and right after fx, so you can simply consider as a 1, okay? Then right after, there's gonna be a transparency function. And how would you define this generalized transparency? It's an issue that we can also make that as a this t of x. And right now, we are not considering about temporal changes because it's always e to the minus omega t, and that's always the same. And t sub of x, you can generalize as an amplitude, okay, times a phase distribution, e to the I phi of x. Here we ignore the like too much of a thingness, but we simplify in this way. So I hope this one will make you a little clear that we can generalize this condition of transparency right after. So this transparency, in fact, is what we call distribution of a sample, okay, in space. So now, I want to discuss about the contrast generation in microscopic imaging. So let's get back to the question of, again, what is imaging? So we are, this course is about biomedical optical imaging. So let's try to expand our concept of imaging. So imaging is a kind of mapping process or mapping function from object space into the imaging space. And we are trying to map some property of an object, okay, that I put it as a physical property. But in fact, it could be physical, it could be chemical. It can be anything, in fact, some kind of property of an object, and we just display into imaging space into a visual form. That's more of a generalized definition of an imaging. Then when we say physical property, or more generalized, I would say contrast mechanism. So what would be those? And I have some illustration of things here. So let's think of what we know about a lot about imaging. For example, light imaging, the same one as I just said, the overhead projector, okay? And the X-ray imaging. So you guys all remember that X-ray imaging is absorption of X-ray to give a contrast onto your screen. So that's a good example. And another thing is I put generalized imaging. For example, what you are seeing me is a room light hits me for every point of my body, and then it scatters. And then it gets into the detector, and that electronic image you are seeing right now. So that is a light scattering, which we already know about it. And a little more advanced topic, which we will cover later, is a fluorescence imaging. And fluorescence is a fascinating one, which of course, the palette of fluorescent protein gave a Nobel Prize, I think it's in 2004, to Roger Chen, and one in Colombia and the other one from Japan. So that's also a very important for the advancement of science, because you can give a special specificity to the molecule of your interest emitting light of your interest, and that can be even colorful. So that color gives you a multiple of information. So this is a fascinating one, and that process we call emission. Okay. So emission excitation, I think we'll discuss this one more later. And let's think about other kind of imaging. It doesn't have to be optics, okay? So for example, you guys all know ultrasound and common photography as well. So ultrasound is sending ultrasonic sound wave. And then when it gets inside our body, mostly our body is in many of the layered structure. So when there's a different layer, when the wave hits, some part will reflect back, some part will be transmitted and hit another layer. So we just listen to the sound, especially the time of flight, then we can actually find out the distance of the reflection and the strength of the reflection, and that gives us a nice ultrasonic image. So where comes the contrast? Oh, the contrast is coming from this wave reflection at the interface or wave scattering with some part of the inside your body. Okay. So another one, you guys remember probably MRI, the mechanism, contrast mechanism of MRI is a hydrogen atom. So that's abundant in the water molecule, and the relaxation property of this magnetic dipole of this proton determines, actually gives you a special information, and that you can use as a contrast, and you can even make a nice imaging of very precious information, especially soft tissue contrast. You can even find out some tumors, very tiny tumors arising inside the body. So that's an excellent way. So all this, of course, another one I want to discuss is a nuclear imaging. That is also very special contrast. The reason is, we are using radionuclides, and that gives a gamma ray, and you put this atom into molecule, for example, a glucose analog, and you inject into the body, and then this glucose analog will spread inside the body, and then especially it will be absorbed by the cells which is metabolically active. For example, like tumors and brain tissue, like brain neurons, or your heart. And those radioactive activity can be detected by a special detector, such as like you guys know PMT with a special forgot to say special detector. So that can detect the presence and location of radionuclides and the concentration. So that converts into an imaging. So all this one imaging, now you are understanding that imaging is a process which really is about contrast. Without contrast, imaging doesn't exist. Okay, so but for the narrowing down in microscopy, intrinsic, sometimes the specimen itself provides a direct contrast. For example, absorption of light. For example, you have a tissue, your blood or your melanin pigment provides a direct absorption contrast, or the scattering itself provides a contrast. You guys remember the light and tissue or light matter interaction, depending on the size of the scatterer, you can consider Rayleigh scattering or Mie scattering, and that gives a intrinsic specimen nature or characteristic as a contrast. And when you're using like fluorescent molecules or fluorescent target, sometimes you have autofluorescence in your biological molecule. And so this could be also quantities of interest. So there are a bunch of this mechanism. Remember, contrast is everything in the end for imaging. So now you can raise up question that, oh yeah, I heard very important quantities, aspects of imaging. One is resolution and the other is contrast. And you can always ask like, what's the resolution of this system or what's the contrast of this image? So that I have a note that the resolution is a property of the instrument itself. So that's why whenever you develop a new imaging technique or microscopy, you are supposed to characterize the resolution. Either it's a spatial resolution or temporal resolution. It can be lateral resolution, or it can be axial or three-dimensional resolution. That only depends on your system. Okay. However, contrast is a little bit different. As you can see from the top, contrast doesn't necessarily come only from your instrument, but it also depends on your sample condition. For example, you are injecting a dye into a mouse vein and try to image blood vessels of the mouse, then the concentration of the dye matters at the end, the signal to noise ratio of your vascular imaging. Okay, so you have to remember, understand the contrast depends not only the instrument, but also the sample itself. So that's a difference between resolution and contrast. Okay, so let's come up with a sample contrast again. So I bring the transparency here. So you can consider the sample as two types of sample exist. Of course, it can be mixed. One is the sample itself has a color, or the sample itself has a absorption, okay, darkness, okay. For example, in this sample, if there exists, this is a good example of a overhead projector transparency film. So you can consider it as a gray of the absorption. So how can we describe it in an easy way? Is let's make a rays and wavefront, and this is flat. Let's assume there's no phase retardation spatially. Then the wavefront will be still the same. However, through this darker one, you definitely have a lower amplitude. So you can see the arrow has a little thinner here. So in that case, this transparency film, you can mathematically describe it as an amplitude distributor, while if there's no phase changes, then the phase function is gone. So then what happens? As an intensity right after this absorptive sample, it's just this amplitude square. Remember here, what I'm describing is an electric field. Okay, I'm not talking about intensity. Intensity has to be magnitude squared. Okay, but let's think about a transparency, which is a good example is biological sample. As I say to you a lot of times, our body, this biological sample scattering dominates compared to the absorption, because many observers are in fact in visible range hemoglobin and the melanin pigment. So otherwise, biological tissue is really kind of a scattering sample, and so it doesn't have a lot of absorption. So in that case, you can consider, hey, I have a cell culture layer. How can you describe it? So you send a plane wave, and through this, you see the arrow itself remains the same, which means there's no change in the amplitude of the light, while the wavefront is distorted. And the reason is the thickness of the sample, the thicker one part will have a more retardation because it has high refractive index. The wave passing through will have a phase changes further compared to the thinner part. So you can describe it roughly this way. And how can you mathematically describe it easily? Is a transparency film. Now has your A of X is just 1 because the amplitude doesn't change. There's no absorption through the sample, while your phase has a distribution. of phase retardation. But the thing I want to draw your attention is we are talking about the field here, but in the end, when we detect a light or a light wave, most of the time we detect the intensity. So that's why we have to see intensity is just magnitude scale of this. And you see magnitude scale of this only phase object is just 1. What that means, that's why it's very difficult with your bare eye to see a single monolayer of cells through a white light or bright field microscope. It's very hard to see. Okay, so let me give you an example here. So good example for absorptive sample is in fact USAF resolution target. So when you develop a new imaging system, this is a kind of a choice that you want to show how small you can actually show. So it's a good resolution target. And that's definitely an absorptive sample, you can say, while a biological sample, a good example, is a transparent sample. But you can see here, yeah, you can see here these cells having a nucleus here. The cell has a kind of a spread out shape. It looks like polarized, but it doesn't give a very good contrast. That's what you are seeing right now. Okay, so for that, I'll go back to amplitude versus a phase object. And this amplitude, I define this absorptive sample as an amplitude object. And because there's a spatial variance in the region to region, and the intensity right after will looks like this. Okay, while this transparent sample as a phase, oops. There's a this will end. Sorry, somehow there's a weird thing happens for Zoom. Can you guys see the Zoom? Let me see. Oh my gosh. Yeah, there is a problem of Zoom limitation. So, so what happens right now? Let me see. Hey, so in 9 minutes, there's a potential it stops. Then you guys re-enter the Zoom, okay? But I'm gonna just buy this.
Okay, professor.
So strange. Maybe the year has passed already. But oh my gosh. Okay, this is not good. So we will continue and then I think we'll finish within an hour, okay? So by the way, are you seeing anything in the screen? Which I don't see my screen. Okay, here, yeah, you guys see the screen, right? Raise your hand. Okay, great. So phase object is a special case of samples which do not absorb or scatter light significantly. I mean, there's always some absorption or some scattering, but it's not that significant. So we can approximate it as only affecting phase of illuminating field and not the amplitude. Okay. But here I want to make sure that we understand standard detectors such as the CCD camera, CMOS camera you are using in the laboratory. They are only seeing the intensity. They will not be able to see phase directly. That's what you have to understand. Then how can I understand the information of the phase? Hey, light has a property. You can convert this phase into something like intensity. For example, the use of interference. Okay, so remember our detectors, PMT, camera, image sensor, most of this only see the intensity information, especially they are like time averaging too. Okay, so that's what I wanted to say. Now let's get into a step-by-step for the microscopy. The first thing I want to share with you is dark field microscopy. So you have a, let's say here, You have a tissue, okay? You make the tissue, a thin tissue. You put it onto the microscope. Under the microscope, you shine it. You will look at like this one, okay? That's a bright field illumination. And however, you can see the fibers over here, but on the empty spot, these are all very bright. The reason is a direct illumination is getting onto your detector. Sometimes this can be not good because when your sample is actually bright as well, then you may consider other way of giving a contrast. So that's on the right hand side. This is called dark field illumination. You see the difference of left and right, especially this dark field is, you can find the background is black. So that's great. It actually looks similar as a fluorescence microscope. So usually background is black, it gives a pretty good contrast. And look at the sample, it gives a more bright. And the reason is the light photons are scattered at this sample and giving it onto your imager. So light scattered by the sample. Now this is gonna be important and not easy to understand at some time. So I want to say that this process is, in fact, when you have a microscope, you're shining the light through the sample. This is when you can see is when you move your objective side so that this direct illumination doesn't get into objective, then you actually sometimes see this dark field illumination. By mistake, I mean. But this principle can be understood by this. It'll repeat, but let me say there's a sample here. There's the objective lens. And there's a focal plane over here that when you shine the direct light, this is the lens, then this collimated light will focus down at this point. And that part is we call Fourier plane. So one F and the other F, and these are conjugate as a Fourier plane, and we'll get back to this again. But remember here, this plane wave will all focus down at the middle here. So a trick over here is, why don't we put a black mask right over here? Then it'll block the DC component of the light through the sample, and then that's blocked. So that's why the background is all black. So that's conceptual. Okay, the idea to repeat is removing, I'm trying to say as a frequency, but not temporal, but a spatial aspect. So low frequency component of an image, we want to remove that. And how can you do that? A good idea is you put an obstruction on the on-axis at the Fourier plane, or this is we call back focal plane of an objective lens. So I'll get back to this one again. All right. So how can you, this is a good idea. How can we implement dark field microscope? And here's the answer. The point is, whatever the direct illumination, that should not go through your imaging system. That's the point. How can you do that? A good way is oblique illumination. Remember, you have a sample. This is actually a viewpoint from the bottom to see. So this is an objective lens, and this is your sample. Remember, you usually have a condenser lens to shine. And if you are shining, let's put a stop over here, then majority of the light is passing through, but it actually goes on the side. On the side. Correct. Then this one is blocked by this system, and it will never enter into the optical imaging. What that means is you have a complete dark background. Okay, so illuminating beam at an angle, which actually is higher than NA. So I want to put this way. So now effectively you make this illumination of through the condenser lens as a higher NA, then you know the NA is the acceptance angle, and it's beyond the objective lens acceptance angle, then it all is blocked. So then those light scattered from the sample, scattered ones can actually enter into the system. So that's implementation of dark field microscopy. And I want to share as a reference for a number of more concepts. This is a reference, so you just think one time, that's enough. So the one I showed you is actually depicted in the left-hand side. So you block a bunch of central light through the condenser lens. How? By putting a dark field stop, and so that this illumination light will only go outside of this objective lens, so that you have a dark background while those interacted light, scattered light from the sample can actually enter into the system. Another way of, hey, wait a second, then there's too much of loss of light. So you're not happy with it, then you can consider another way. Hey, let's make a mirror surface and you shine the light. Oh, okay. Sorry, this is also dark field stop, but a good kind of idea is mirror surface is shine onto another mirror surface, so it goes very shallow angle, higher, and then it goes outside. So this is something you can consider. Hey, okay. So so that was the concept of dark field microscope. This is pretty easy, but kind of fun to consider, like how to keep a nice way. So this will be helpful when you are trying to analyze the data. So bright field microscope, having a bright background, and you have a sample scattered light adjusted for that. So when you are quantifying, only carving out the sample, it might not be, it could be a challenge, but for bright dark field microscope, you kind of remove the background as a dark. So it provides a relatively better contrast, and it's easier when you have to analyze only the sample, let's say the size of the cell and segmenting the cells you are trying to understand. So in that regards, dark field microscopy is pretty good. Let me wait a little bit for Sadaf. See. Okay, should clock. All right. So I guess Sadaf and Shukla is now on board. Let's move on. So the second topic I want to discuss is the famous phase contrast microscopy. And you see the title, you know, phase is very important in optics and wave, but unfortunately our eye and our camera do not detect the phase directly. That's a problem. So that basic concept, without giving an exogenous dye or killing the sample, there's a live cell imaging which gives an excellent contrast, was given by Fritz Zernick, which is right over here, and that conceptual change gave him a Nobel Prize in 1953. So this is a good example that I want you to always think out of the box. And right now it's kind of obvious, but at the time this was a revolution. So why? Look at these images. So a conventional one's bright field microscope or wide field microscope looks like this. And after his trick, you can actually make this image into this. And what are the differences? You can see something, what's going on, a lot inside the nucleus, and even in the cell cytosol, there's a lot of process going on, which is much clearer than this. And this was achieved without disturbing the sample at all. So that gave a kind of revolutionary impact, and that gives him a Nobel Prize in Physics, 1953. Why? Because it's labor-free, you don't do anything, and so it's non-invasive investigation. So this was a revolution. But you will realize my formal letter signature, I have this quote. So I want to read this one for you. And I think this is something I expect you guys always think like this. and to become a real inventor of something important. So, but that's something I want to share it with you. So what he said is how quick we are to learn, that is to imitate what others have done or thought before. So while we are studying, we are learning what others have done. So the first thing, of course, is imitate. You imitate things people have done. You imitate your boss, okay? How they show. You imitate someone or in the laboratory or your many of the faculty members, you imitate first. But you have to keep thinking because this is a quick and easy way. But how slow to understand is to see deeper connections. So you always think below that, how does this link to the other things, the connections? That's slow, but it's more important. Of course, when you are trying to make a publication for your graduation, but like for science and for the fun, you always try to see the deeper connections. That's how to make your product as a shareable with the community and the other people by trying to see deeper connections. But the slowest of all, in the end, are we inventing a new connections? So that's something you are trying to make all the time, or even in applying old ideas into a new field. What that means is, it's very old idea in one field, but when you are trying to solve a new problem, and you can bring that concept into here. So that's something slowest, but the most valuable and recognizable. So for that, I would like to say to you that, hey, new connections. Oops. Let's see. Are you seeing your screens now? Okay. So there's no problem, right? So that's something I would always expect you. And I think what you have to strive yourself is just imitating first. Of course, that's fine what you are doing. Second, always trying to find out, understand deeply. That only, I think, is possible by keep asking yourself, do I understand it really? You know, I mean, this lecture is kind of bombarding your information, but I want you to think. And in the end, I want you to invent new things. The easy way is applying old ideas. What that means is that's why we have providing you this course, broad course, and so that you can find out those ideas into new field. So that's what I would expect and want you to actually pursue this aspect. So now let's get into this Nobel Prize winning idea one by one, the principle. So this is, or you already have done like 4F system geometrical optics. Now we are getting into a realm of Fourier optics, which will start this Wednesday. So, but this is a kind of first easy way. So let's consider the plane wave gets in here. This is object plane, okay, and then it keeps going majority of time. If the object is like sparse object, and you hit this first objective length, and plane wave will focus down. Make sense? And then there's always tube length, and then it gets onto the image plane. So if we assume the sample as a phase object, so this is typically what will happen. And I actually get this edit this new picture for those who may not have a clear understanding of Fourier transform. But you guys know that, okay, it doesn't say. A constant Fourier transform is 정면. 정면 is not Brahanu. What's the Fourier transform of a constant? Sorry, you are muted.
What's the question? The cosine or the constant?
Constant. Let's say 1. So fx is 1. when you Fourier transform, what will happen?
Mm, I think zero,
Excuse me?
zero,
Sorry,
Yeah.
I can't hear you.
0, 0.
Okay, Sadaf. Sadaf, I can't hear you. We can't hear. Yeah. Sadaf,
It will be mean to say f of t plus c.
okay,
Function of that, I don't know how to explain.
okay, so don't worry. You have to resort to your intuition first before even considering even mathematics. Look at this. Object plane, the intensity is constant, okay? And one lands at the focal plane, and that focal plane, you see what happens. The focus is down. There's only light at this point of 0, while the other is none. So this is, we call mathematically delta function. Okay, so constant Fourier transform is a delta function. If you feel you're not very familiar with this, you should resort to a basic Fourier transform. Okay, there's only a couple, like not even that many. So that's why we are doing with length, we are doing Fourier transform. Okay, constant becomes a delta function. And again, this happens at the focal plane, at the object and lens, and the back focal plane. That's why we call this as a Fourier plane. Does it make sense? And then let's go back to our 4F system. Then there's a tube lens. Okay, so this is another F and another F. That's actually image plane. So in fact, the image will, in mathematical term, you can call image as an object plane, Fourier transform one time and twice. So it's like Fourier transform twice, you will go back to the same. So that's why image plane is seeing the image as an object. Although there's some limitation that you learned from the previous diffraction limit, that we are not getting all the information. That's why our resolution is limited. Okay, so that's the basic concept of the principle. We consider 4F system as a microscopy, and then let's go back. I was talking only about illumination light, but I didn't talk about the light scattered from the part of the sample, which is our own interest and what imaging is about, right? So look at that. Red one is a plane wave, but now we are only focusing on the scattered light. So that I depicted it as green. Look at that, there's a cell. From one part of the cell, you know, the light scattered out. When I define imaging itself, that scattered light is now from the distance of F from the object lens. What's happening? So that scattered light is making a divergent beam onto your lens, objective lens, at the distance of focal length. What happens? They will be collimated. Do you agree with it? Right? From your geometric optic. And then through this tube lens, because it's a parallel, they will focus down back. And this is your image plane. So a point is mapped onto the point. What about the side point? From that point will be pointed on this side. So that's what we call imaging. And here's the deal. And now, why don't we try to make this as a simple mathematical formulation? Okay. So the field, let's focus. we are dealing with electromagnetic wave. So we have to think through the field first, but remember the imaging process, we are only seeing the intensity. So at the latest, the field magnitude square gives an intensity distribution that we call as an image, okay, recognizably. So let's see as a field right after sample. Let's assume the sample is a phase object, such as a biological sample or a cell. Then you can consider it as f of xy laterally, and the amplitude is gone, so it's 1, it's constant, so e to the j phi, so it's only phase object. And here, I want to draw your attention from your high school Taylor series expansion that e to the x is 1 plus x squared over 2, okay, plus blah, blah, blah. So if the x is actually small, you can actually remove, you can neglect the higher order term. That you remember that, let's just apply, because this phase object, the phase distribution is not that great, okay? So assuming that one is holding, then this field right after the sample can be described as 1 plus j phi. And let's put the meaning of it. What does this 1 mean? It's like there's no sample, it's like constant. So that's a, we call it DC component or unscattered light. And the j phi is a phase distribution, and that we call a scattered beam. Okay, so then that's field right after the sample. What's the field on the image plane? Because that's supposedly a map of this sample, so you will have the same as a field at the image plane, 1 plus j phi. It's an approximation. But we are not seeing in field directly, we are seeing only intensity. So that's why intensity is magnitude square. And I would say the phi originally is smaller than 1. So this is 1. That's why in general bright field microscope, you are only seeing a roughly kind of intensities are similar. Only there's a slight variations. Okay, that's something why we see phase objects such as cells are almost invisible at the imaging plane. So this is a little bit mathematical description of what's happening in general microscope. So now let's think of this in another way to give a new ways of contrast. So how? Yes, you know, there's some, why don't we do some mathematical trick that let's introduce something like phase object at the Fourier plane. In this case, we saw that something's happening. So at the center, maybe we can do some trick. How? Because we know field after the sample is e to the j phi, and that's approximated as 1 plus j phi. Okay, and as I said, this and Fourier plane and the imaging plane, the relationship at the Fourier plane is Fourier transform of this sample plane. So from that aspect, as I say to you, hey, field right before the phase object, this Fourier plane, let's do the Fourier transform of this. And you guys now know Fourier transform of constant 1 is direct delta function. Okay, those who do not understand what's delta function, please resort and even see your TA or me, you can just Google it, okay? And the other is, let's say phi, Fourier transform as a capital phi. The only thing is the xy becomes now spatial frequency components, so-called u and v. And why don't we give a trick? So you see there's a j over here, okay? And you know that this one, sorry, this is a u comma v, and but that's only having component at the center of this part. So looks like there's a j there, and we suffered from j before. How about putting a phase plate which shift by 90 degrees only at the center? And what that means is only here's, these are like broad, so it doesn't care that much, but that one's only happening at the center. So we give center just a phase shift 90 degree in Fourier plane that we can actually convert just multiplication of J. So making it imaginary. So that's right after field. So you image plane, you can Fourier transform, and you now see this field at the image plane as a J of this. Because you are doing inverse, not inverse, Fourier transform again, and then you will say have a J together. Now you have the component 1 plus phi. Okay. So remember, you have on homework of reading the textbook, especially that Fourier optics part. And if something's unclear to you, you have to go through. Okay. So now let's look at what happens to your image. And that's, you have intensity, which is magnitude square, then this J is gone because it's common to all of this. And 1 plus phi magnitude square is 1 plus 2 phi plus phi square. And I say phi is the phase, and that's smaller than 1. Square is negligible. So what you get now is very interesting. You're in image plane, you have intensity not only almost like constant, plus now the phase, twice of the phase become an intensity. So this is the concept to convert a phase object into an intensity object. So does that make sense? So that gives these images. And we can say enhancing the contrast of phase object by use of phase delay. Okay, so go back to the mathematics again, that what's happening in this bright field, this is almost constant intensity as a 1, and the other, you see something very interesting variation. And even you can see sometimes a halo on the backside of the set. Isn't it curious? And that's, I think you can also consider from this mathematical description, as bright field is intensity is 1 plus J phi magnitude square, and that's a real number, and that's close to 1. That's why it's almost kind of uniform light, while this one is 1 plus, and this phase become converted into an intensity. And here comes actually your homework problem. So I want to generalize this trick into a little more general form. So you have a sample, you have a plane wave coming in, an objective lens, and Fourier plane. And the trick, what we say is, before we just have here as a phase object. And then we now know sample Fourier transform into this Fourier plane, and another Fourier transform onto your image plane. Why don't we generalize this object and add the back focal plane as more generalizable, as let's assume there's a general amplitude times e to the j alpha, which is our phase retardation. Before, for this phase contrast, we gave this e to the j alpha. Alpha is pi over 2 or 90 degree. So that comes as a cosine 90 plus j sine 90, which become a j. So that's how we convert. But if you think about it, you can more generalize as A as an absorption. So what does this actually mean in physical aspects? It's placing a small metal film covering the DC component in the Fourier plane of the objective to both attenuate and phase shift of the unscattered field. And that's the trick, and I think you want to think through, and that's your homework. Okay. And you see, in fact, if you are blocking this as just a block at the middle, what happens is the DC component is gone. And you realize that's a dark field microscope. And if you have this absorption as a kind of a function between 0 to 1, you may be able to find a right one to give the best contrast on your image. That's your homework. Okay, so how can we implement? I gave you a very simple one-dimensional or two-dimensional idea, but let's try to implement. Then you want to consider it in a more general way. So look at this, because there's a sample, there's a condenser. This is actually 2D cut-through version of real 3D. So you have to interpret a little bit carefully. So let's look at this light source, kind of point light source. Then remember the parallel illumination, you should shine the sample in a parallel. And then this parallel ones will focus it down over here. So that focus down, you want to put the proper phase retardation to convert the phase information of the sample into the intent. So see this red one and become converted onto your image. Just likewise, the other side is doing the same. And you do this kind of a circularly. That's the concept of how to implement this as an annulus illumination. And at the conjugate plane of this part, you put a phase plate. But as I say, this plate is the objective lens backfocal plane. Typically, objective backfocal plane is inside the objective, not always 100% most of the time. So that's why I brought a phase contrast microscope. Requires a special objective lens and condenser. So this is the reality, how to make it. The phase contrast microscope, the condenser, and over here, there is a condenser annular. So remember the microscope I showed you previously? In fact, there's this ring over here. It shows a pH2, pH1. Let's take it out. I'm going to show it to you. Look at that. Oops. You see, there's a annulars, two annulars. There's a two things because there are two different kind of objective, pH1, pH2, depending on that. That one's linked to this condenser annulus. Sorry, I hope this one is obvious to you. And then what about this phase plate? So you have to pay more money or buy a special objective. As you can see, it says pH1. So this is phase contrast microscope objective, and it has a special phase plate at the backfocal plane. So let me try to show you that part. Can you see the circle inside? Yeah, that is giving a 90-degree retardation at the backfocal plane, which is conjugate to this pH1 ring. So with this, you can have an excellent imaging of this. So the trick is phase contrast microscope do not require any more complex things. You just need this condenser annulus, and you use a phase contrast specialized objective lens, and that gives you directly this nice images, which is normally you cannot see this high contrast because it's all phase object, like such as RBCs and some Chinese ovary cell, typical cells, and you cannot see this high contrast, and this is fish scale. So now you learned all about bright field and phase contrast microscope, and I'm going to introduce you differential interference microscope and later fluorescence. But I want you to actually, when you look at the image, you should be able to now be able to say bright field and phase contrast. Phase contrast is annular, so that means it actually affects, there's no orientational preferences. However, the next one is very interesting. You look at this differential interference microscope. Look at this one, is almost like shadow images of a 3D object. So this is also very, very cool because it just gives a very interesting way of giving contrast. But remember, there's one directionality of the shadow. So that's something interesting. Fluorescence we'll discuss later. for that, we want to discuss a little bit about a polarization. Let me come back to you that electromagnetic wave, plane wave propagation onto Z direction. We freeze the time. This is what electric field of vertical polarization. So if I froze the time at Z equals 0 and always at Z as a lambda, it's coming back. Okay, but what about this is only vertical electric field? It can be horizontal polarization. Okay, so these two are general because in the plane of incidence, your electric field is somewhere in between vertical or horizontal. You can always decompose into these two components. And now let's think about more general, that is electrical, elliptic polarized plane. And that's very generally, the horizontal and vertical has some phase shift. What will happen is if you do this like geometry of drawing, you will realize it actually is a changing in space. The electric field is changing over the space. So that becomes a elliptic polarization if the amplitude are exactly the same and the phase retardation is exactly lambda over 2 or 90 degrees, that you will result in circular polarization. So circular polarization is a combination of horizontal and vertical field, just about the right retardation of phase shift. And to help you understand, I give you this circular polarization movie. So this is what's happening over time and space of electric field. So if you are actually looking at Z equals 0, you see this kind of changing a big arrow. So that's what you see. So at space in one place, you will see this circular polarization. But in space, this is what's happening. And I want to give you an idea. What if we freeze the time, this is what you will see in spatial distribution of electric field. And you can decompose into vertical and horizontal in a lambda over 2 wave. So like a half wave is a phase retardation between the two. So that's why it looks like this. Okay. So in general, circular polarization can be having linear polarization sum of vertical and horizontal with a, okay, sorry, this is a pi over 2. This is flipped. And now I want to give you an idea. You guys learn about linear polarizer. I actually have a sunglass, which is a linear polarizer. And what does it do? Is it passes only one direction, so it blocks the other. Hey, then what can be a problem? Because if you have high power laser, this blue part is absorbed by this polarizer, so you can burn the polarizer. So be careful if you're using hundreds of milliwatt or watt, you cannot use linear polarizer, or you will be able to burn and ruin your polarizer. Just remember that. So for that, there's another interesting one called PBS, or polarizing beam splitter. And that actually splits into different polarization states. Normally we call SMP polarization. So depending on this polarization, you actually split. And sometimes you actually combine them together, which is a good trick. And this is a good way of control light power, because the input and the output, you can actually take off the other polarization. So if you have something polarization angle changer here, such as halfway plane, you will be able to control the amount of light. Okay, and there comes a very interesting calcite. So I think I want to introduce, for explaining DIC, I want to introduce a specialized prism, which has a property of like spatially separating different polarization state. So how? By use of this special calcite, which looks like this. You see, in Korean, it's called a 복굴절. Okay, so, or in English, it's called birefringence. It's optically anisotropic, but it's transparent. But you look at this, depending on the polarization state, you can see two letters, which originally is one. how it happens is you can see different polarization actually are split into different direction. Now I have 5 minutes left. So that's the concept. The concept is, hey, how about we can separate two different polarization states in space, and then we put the molestin again to combine them. And why is this good? Because you separate in space, let's say there's a cell over here, and you separate the beam this way, one goes through the cell, the other goes to free space. Then what's the difference? You are imposing a phase shift through the cell, and if you combine them together, you are interfering, okay? So what happens is, if you separate, you go through the center of the cell, they suffer from the same phase retardation. There's nothing else. What if the edge, one passes to cell, the other passes to free space? This phase change, now you combine them together, you are effectively generating interference. That's the trick. You separate them onto the sample, and especially at the edge of the sample, these red and blue will have a phase shift, and you combine them, that means interference. So that we call polarizer and analyzer here. So how we can make it? By making first a defined polarization and sending to Wollaston prism, and that light goes through the sample and put another Wollaston to combine them to generate interference. So the cool thing is now again, there's no special stain, okay? And with a full NA. So this is the mathematical description of differential interference microscopy, that Wollaston prism separates them from at the sample, and then you put them back together so that electric field is combined, but one has a spatially displaced on the sample. And that's not a lot, just only about 1 micron. And when contrast comes is when this E1, E2 passing neighboring path on the sample, especially one is sample, the other is outside of the sample, you will experience a different optical path length or different phase delays. And that's the trick. And let's make it, it's a little bit of mathematical formulation. Then let's say now you have this Wollaston prism by changing or adjusting this through the shear axis. What I do is I'm imposing some of this delta of phase change for this e to the 1, e sub 1. So E0 and e sub 1 now experience different optical path through the sample. And by applying this shear axis, it only applies in one axis. Let's say that axis, this axis is an X, then we'll impose a phase delay, phase change from the sample. And also by displacing the position of the Wollaston prism to add an additional, sorry, this might be a little confusing. So this X and X plus delta phase, these are coming E0 and E1. And by shearing or displacing Wollaston prism, we are introducing the E to the 1, an additional phase difference, and which we can adjust to make the higher contrast later on. And how can we do? Because electric field is summation of this, and your intensity is now magnitude square of this. So look at this mathematics, e to the phi x plus delta, and introduce Wollaston, and intensity at the image is square of this, and you will get this one. Okay, then you will realize phi is more, and this introduction of delta is a half or 90 degree. Then what happens is this cosine, because you know from the trigonometric formula that this becomes a sine of this formula when this becomes pi over 2. And you know that sine, when these are very small, it can be just about approximate the difference, and you put differential over here. So now what you have is your intensity of image contains this differentiation onto x-axis. So the final image is now giving phase gradient is shown. Let's understand what does this mean. Is here, now you have some axis in here is an x-axis. So you can see clearly there's one axis. If you look at the intensity, optical path difference, because RBC has a higher refractive index. It's okay. Okay, so here's the deer. Look at that. By very careful introduction of a Wollaston prism onto x-axis that we call shear axis, the result is quite interesting. Now you see intensity, not just this is a kind of phase changes, which you cannot see in normal way, but by introducing this, separating in space and combining after interference, you get this DIC, and why we call differential is because differentiation is coming into your intensity. And if you know from the math, let's say these are phase, if you differentiate this, then you will get this one. And that's through this axis, what's happening, the high and low, high and low. So now you have a very interesting, like almost shadow-like imaging without touching the cell or sample at all, because now you have this gradient. But look at this as a one axis. Now we've learned everything for today. I want to give you the images of various contrast mechanism, and you should be able to tell, look at that. The upper ones are phase contrast. It doesn't have orientation, but has an excellent contrast from the phase, you can see. And exactly the same sample, if you are using differential interference contrast microscope, unfortunately, this one didn't get Nobel Prize because usually it's given the first groundbreaking idea, but it's also very fine. You can see a nice, like three-dimensional shadow type images out of this. So the two differences, you can see the presence of halo and the shadows, and this is all like contrast enhancing with no labor. Let me tell you the problem, and always you have to focus on still what are the issue. You're not quantifying things. You are giving a qualitative enhancement of contrast, but you're not actually giving a number. So that's the differences. And here comes the deer, this phase contrast now. You know, I'm telling you the state of the art. I think it can be a part of our later on, we can see. Now we can talk quantitative phase imaging. So this phase information, now you can quantify what that means. You can actually measure kind of a cell size, or you can convert in cell volume, or even protein mass, something you can make it more quantitative. So that's an emerging modality, I would say. All right, so now we are done, and I want to review with you with these 4 ones. So remember, the first one was a bright field. Second one is dark field. Oh my God. The third one I didn't cover with you. Okay. Fourth one, very interesting. It looks like kind of differential. So let's go back to one by one. So what are the contrast mechanisms? Let's turn on all your audios. Okay. Let's go one by one. Let me ask Brahanu, what's the contrast mechanism of bright field?
Light illumination.
Okay, great. Yes, absorption. Sadaf, what's the contrast mechanism of dark field?
Reflection.
Excuse me? I give already one. What did you say, Laxman?
Sorry, bro.
Scattered.
Okay, yeah, Brahanu. Yes, light scattered by the sample. Great. Let's go back to Sadaf. Phase contrast microscope. What's the contrast mechanism? 정묘? So, phase we cannot see directly. That's why we convert the phase into a intensity. How can we do that? Anyone else?
Interference.
Okay, coherent. Okay, ding dong ding. Great. That's the interference. Interference makes this phase into the intensity. Yeah. So now get back to the most difficult question, which I didn't even cover. But look at that one. This one. This one now is a kind of, I didn't cover it that much, but remember there's a polarization. So you can have one polarizer and you can do a very easy experiment. The other polarizer is 90 degrees. When you shine the light, you will have it all like blocked, okay? However, if there's a sample which somehow changes the polarization somehow, and that light can pass through. So that's something I wanted to show you. Okay, I don't know where did it go? So yes. And so polarization, if remains the same, it will be all black. So it looks like dark field, but this sample has special property to change the polarization so that they can be able to pass through this kind of orthogonal polarization. So anyone can answer this. I think that would be like really great. Any guess?
Refraction.
Refraction. Refraction is kind of change of direction. It's kind of close, but we are talking about polarization. That's a direction of electrical field. So if polarization doesn't change through the sample, it will be all blocked because you have this polarization coming onto sample and then the other orthogonal polarizer and you have an image. So nothing will show up, but now the sample somehow changes the direction of polarization. How can you say for that?
Direction.
Rotation.
Yes, rotation. Rotating, somehow the sample rotate the polarization that will actually be able to go through the next polarizer. So this is rotation. Okay, I think you guys are now expert in microscopic contrast. I know some of the concepts are quite challenging too, but it'll be more clearer once you learn about Fourier optics from this Wednesday. I have to say the Fourier optics is the flower of graduate level because from there a bunch of Nobel Prize winning ideas are all came out from there. So but we don't have much time, so I'll put the previous, like 4 years ago, a previous lecture note first because I want to update it, but I'll give it to you so that you guys can actually go through a little bit. That's going to be the most tough, probably content in probably in this class, and tough, but the most important. So let's summarize. Today, we discuss about contrast mechanisms on imaging. Imaging is about contrast and whatever property of physical, chemical, or even like something very weird thing which we didn't even have the sample, for example, radionuclides, something which gives a contrast. That's how we can make an imaging recognizable, quantifiable, and analyzable. And for that, we went through the history of a novel ideas of because we cannot see our biological sample, which is too transparent, which is not normally an absorptive, but more like phase object. So we engineered a new ways of converting this phase information with a mathematical trick or physical trick of converting it into using interference somehow so that electric field, which can be converted into an intensity. That's a phase contrast, and dark field is another key aspect of, hey, let's remove all the background, which is DC component, by giving a trick that understanding of Fourier plane is a Fourier transform. So you know where the DC component ends, and let's just block that part only, you'll get like dark field. And another way of implementing it, just make an oblique illumination so that the illuminating light never gets through the optical system. So you have like a nice sky dark background. And understanding of the orientation of electric field that we call polarization. And that polarization property of light is very special. And so that we can actually utilize polarization specific ways into our imaging, which is kind of something different from sonic wave, ultrasonic imaging, because sound wave doesn't seem to have a polarization, while electromagnetic wave has polarization. And that linear, more generalized elliptical or circular polarization of this plane wave gave us another trick and tool to develop into a newer ways of imaging. For example, like introducing Wollaston prism of separating polarization and combining, and through the samples, optical phase difference, converting into an intensity as a differential. So that's a very, very cool technique. And here comes that DIC microscope. And we now try to understand this one into a little bit more mathematical and in terms of Fourier transform aspect. So you will see like even light can do Fourier transform itself. I mean, they do the math, the nature of these optics can do the math by itself. So with that, I want to summarize and thank you for all your attention. Okay, so I shared this journey. This is an essay, you know, read it for fun. I also even read and make a line. So it's always exciting to see, you know, it's very easy for us just to follow what has been done. But the first time inventing things are different. And I hope you kind of, you know, read this and feel the grasp, the gist of how the discovery was being made. And that lecture gives his discovery on phase contrast. And for more of Fourier optics aspects, I shared with you the book chapter of Gabi Popescu, one of my good friends who used to be a postdoc when I was a graduate student. He is now the leading pioneer of how to make phase imaging into quantitative. And he was a very good teacher as well. So he wrote a book and I shared it with you under the permission of him. He generously gave this file to me. Otherwise you have to buy the book, right? So that's all. And also these websites are useful.